Sean Carroll, thank you for joining us.
Thanks for having me, Nick.
It's been a long time for the listeners who don't know, which is probably everyone. You're actually my scientific grandfather, I guess. I got my start in science in your lab.
I remember when you first came in. You were one of perhaps the only incoming freshman I ever experienced who had read, and you'd have to tell me how many of my books you had already read. And I thought, my God, I can't get my graduate students to read these books. Here's somebody who hasn't even taken his first freshman class, and he's ready to rock and roll.
Yeah, it was your first book, and we can get to that. I definitely want to talk about that book, but you're really interesting because you're a biologist, but you're not just a biologist. You do a number of things, and so how about if you meet someone at a cocktail party, they have no idea who you are, and they say, "So, Sean, what do you do for a living?" What's the first thing that you say?
Oh, I tell them I'm a biologist. I think the science identity is foremost. It's kind of everything revolves from that. And I've been really fortunate in the course of my career to have periods where I focused exclusively on what I'll say is discovery science when you were in my lab. Um, I was able to branch into writing sort of somewhat at the same time. And then more recently into filmmaking or being part of a whole filmmaking enterprise. And then areas of philanthropy and all this other sort of stuff maybe we can get into. But it all started with a scientific lens, a scientific perspective, and both the desire and the opportunity to share that process, the substance of those discoveries or the journey of those discoveries, and sort of the scientific worldview with all sorts of audiences. And I really enjoy it. And it's just sort of diversified into lots of different media, whether that's doing, stand-up talks and live audiences or books or films.
Hmm. So, as a biologist, what do you study? And are you still running a lab right now?
Yeah, so I have a lab at the University of Maryland. So for the last good amount of time, it's been exclusively focused on evolutionary questions. And the big question my lab is interested in is where do new things come from? And that's also something you bit off in your undergraduate work. So the focus of study has changed because you attack different use different models that you think might give you insights into certain questions. And so in the early days, I was really looking at new physical patterns. For example, body parts and body patterns. Where did new things come from there? More recently, wanting to know where new, for example, biochemical functions come from, and particularly using snake venom as a model.
Hmm.
So venoms have evolved many times in the animal kingdom. Fundamental question is, well, where the heck did all this stuff come from? And to what degree is this all new stuff or old stuff that's been repurposed?
Hmm.
And fortunately, we're living in a time where those kinds of questions we can get down to the brass tacks of how things work.
So, when you study evolution, let's just take the venom example. How the hell do you even study something like that? What do you guys do?
Well, you start with the stuff you're interested in. So, let's take rattlesnakes. We chose rattlesnakes because we knew we were going to have access to material, and there had been a recent fair amount of study of them. And venom is this cocktail of proteins that the snake injects into its prey or accidental human victim. And these things do a bunch of stuff that essentially take the prey down, either kill it quickly or at least disable it so that it can be consumed. And because it's a cocktail of stuff, you'd ask, well, how did it get all this stuff? It couldn't possibly get all this stuff overnight. So it's been assembled over time. So how do you figure out how this was assembled? And where did it come from? And when you say where did it come from, proteins are encoded in DNA. And so that means DNA is really the crucial record of invention. And what you want to do is study animals that have and make venoms like rattlesnakes and compare them to things that don't have those things and say, all right, here's a certain toxin gene, where the heck might it have come from? And you get clues from, for example, if you sort of parachute into the DNA where that toxin gene is and you look around, you might find adjacent genes that are present in all sorts of non-toxic animals. And that starts to give you a clue of where that gene came from. And then you look at that gene in detail and say, well, does that look like an old gene that's just being used in a new way? or has the gene been modified in some ways to maybe, sort of weaponize it? And so these are the kinds of questions that we're asking. So it's a comparative approach, and then it's a deep dive into sort of the DNA record of evolution to say, where did this arsenal of toxins come from?
Hmm. Well, so in the case of rattlesnake venom, what are these proteins? Do they look like other proteins that do something more basic?
Absolutely. And they come from essentially household, housekeeping proteins that
Hmm.
all of us have. Uh, Very familiar stuff from a lot of four-legged animals and fish have. So, for example, in rattlesnakes, two big classes of toxins. They have these toxins that are, certain rattlesnakes have toxins that are neurotoxic and that can drop you pretty quickly,
Mhm.
respiratory arrest. Um, and those came from a class of enzymes called phospholipase A2s. And they've been modified a bit, but nonetheless, they're totally recognizable as members of that family. And there's another group of proteins. These are enzymes called metalloproteinases. It just means they chew up protein, and they use a calcium or zinc as um, in doing that. And we have tons of them. Lots of animals have tons of them. And just one of those was co-opted and modified a long time ago in this lineage that gave rise to rattlesnakes and their close relatives, the vipers, and some of their more distant relatives. And the snakes have just gone crazy with this. So one gene that you can see in all sorts of four-legged animals has been expanded to a battery of 30 genes in the Western diamondback rattlesnake.
Hmm.
Bam, 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 right along the chromosome, you see it. And that arsenal, that battery of toxin genes is telling you something has been specifically expanded in this lineage that is obviously important to the way it lives. So those are the kind of forensic clues that we can discover in DNA that tell us where the toxin genes came from and how they've been modified. Now, we didn't know any of this 6 or 8 years ago because
Mhm.
we were the first to look in the genomes and ferret some of this out. But nonetheless, we've been surprised lots of times. First of all, it's gratifying to be able to trace the origin of things. That's not something that evolutionary biologists have been able to do until fairly recently. But also the snakes they do some weird things. There are snakes that there are populations of, for example, rattlesnakes in the Southwest where some individuals are neurotoxic and some individuals are hemorrhagic. And there's really surprising levels of genetic variation between individuals. We know that ancestors of most rattlesnakes were neurotoxic, but a bunch of species are not. And you think, well, once you invented a neurotoxin, why would you ever get rid of it? But that's
Mhm.
clearly what they've done. So, as has always happened throughout my career, you get surprised because it turns out the real story is opposite or upside down to what you expected. That you would think when you invented a potent toxin, you'd never get rid of it. You'd think that if it was a really potent toxin, every member of the population would have it. It's not the case. So we're enjoying kind of the natural history of these snakes at the same time understanding these kind of general evolutionary questions that we're trying to pursue to a real satisfactory resolution.
Where where do you get the raw material?
Yeah, so we have collaborators in Texas. A woman named Elda Sanchez runs a big center there that houses all these snakes, and it's a federally funded center that houses all sorts of poisonous snakes. And they've hosted my lab group multiple times. We also get stuff, through FedEx. Um, I can tell you that when I moved my lab from the University of Wisconsin to the University of Maryland, my Maryland hosts were quite happy to hear that I was not bringing tons of rattlesnakes that they would have to house. Because you can imagine on a college campus that bad things could happen if you had a basement full of rattlesnakes somewhere. So the material is pretty straightforward to get. A little bit of venom goes a long way. A little bit of blood for DNA goes a long way. So um, yeah, and now some of those tissues we can culture, so we don't actually need to take things from living snakes. So it's sort of becoming a little, it's not quite fruit flies in terms of
Mhm.
ease of, certainly not ease of housing or anything like that, but in terms of experiments and the kind of studies we do, it's opened up. I should also mention, people may not care at all about snake venoms, although they're a pretty interesting model. But think of parallels. For example, the invention of milk
Mhm.
in mammals or the invention of antifreeze in certain animals. These are comparable stories of, well, where did new things come from that sort of open up lifestyles? So that's the general question that we're trying to understand is, how do things get invented that then sort of blast open new ways of living? And rattlesnakes themselves have radiated fairly recently to a really good number of species and their close relatives, which are called crotalids, across the New World. So there's been a lot of action in snake evolution in the last, say, 12 or 15 million years.
Hmm. So rattlesnakes are interesting. You also mentioned fruit flies, which was the animal that I worked with and that you worked on for most of your career. One of the things I learned early on in your lab and elsewhere was to say, we didn't study fruit flies. We studied something deeper and bigger, and the animals were just a tool. So what were those bigger sets of questions that you were using them for?
Yeah, great question. I think just a really important question when talking about science. Look, I think the art of this business is you try to find the simplest example of the phenomenon you want to understand. If you try to find the most complicated example, let's take something that I've never worked on, like the brain, but you
Mhm.
have. And you start with, an elephant brain or a human brain, you're dealing, it's complicated and it's hard to do experiments. So you move to something simpler. Well, in the case of the fruit fly, this tiny little animal, really in the building of that animal and its wings and its eyes and its legs and everything else, it's really just a microscopic example of anything that any other animal has to do. But they're very easy to keep. They reproduce rapidly, and because they've been sort of the workhorse for genetics for a century, there's just tons of tools that biologists have come up with over the decades.
Mhm.
So, I didn't say to my parents, "Oh, I want to be a fruit fly biologist," or something like that. What happens is you find models of what you want to understand, and the fruit fly turned out to be a great model for animal development. So, how are complex things built?
Well,
And then go ahead.
why is that? How can a fruit fly teach us something about other animals?
Well, it's now an answer I can give more in hindsight, because the honest truth was in 1982, when I was deciding to go after my PhD and go study fruit flies, I had a few of my mentors say, you're stepping off the edge of the earth, because there was a bias in the biology community at the time that if you wanted, you had to study something relevant to humans, and that would mean studying animals with fur, like mice. And there was no expectation that something like a fruit fly, which is an arthropod, an invertebrate, would be a model of anything for humans. But but there was so much fascinating stuff going on in fruit flies. So the phenomenon that launched 1,000 postdocs was the discovery of these mutations called homeotic mutations. And they are mutations in single genes that transform one body part into the likeness of another. So antennapedia, for example, transforms the antenna to leg, or a mutation called bithorax transforms the hind wings to the forewings. So you get a fly with two sets of wings instead of one. And
Wait, wait,
the
wait.
fact that,
So you mean literally a mutation in a gene will turn the antenna of a fly into another body part?
yeah, into a leg, to a nicely formed leg.
Hmm.
And you think, man, I got to understand how that works. So when I first read about these mutations, 1981 or 1982, I was studying something completely different. I was getting my PhD in immunology. And I thought, what could those mutations be? What could those genes be? There, a single mutation could so transform the body of an animal. And they seem to have some sort of evolutionary significance, because you sort of think, if a gene can really sculpt a body part like that, well, what makes a lot of animals, interesting difference between a lot of animals are the number and kinds of body parts they have.
Mhm.
So I was hell bent that I was going to study genes like this. And it was not really on the radar of many people at the time. And I approached a really young guy, in fact, somebody, Matt Scott, who was just opening his lab. He hadn't even opened his lab yet. And I said, hey, I want to work on these genes. And he said, okay. But, first day in the lab was cleaning out the glassware of the lab, the previous lab that had left it all messy and dirty. We really started from the ground together. But it was a good call. Again, wisdom was the wisdom I was getting was this was a really dangerous and risky maneuver,
Hmm.
but I was driven by the biological interest. I just thought these mutations were so fascinating. The genes must be interesting. There wasn't There weren't a lot of tools for getting at genes in those days, or for figuring out how they might work inside an animal. So even when I wrote for a postdoc fellowship, there was a lot of skepticism of whether or not this was even technically feasible to figure out how these genes worked. So you have to kind of, sometimes you have to swim against the current and say, look, this thing is so compelling, so interesting. I gotta know, I gotta work on it. And fortunately, I stuck to my guns, and then I got lucky. And I was in a great environment as a postdoc. It was really fertile. It was really creative. A lot of things happened in a short period of time. And to get back to your original question, why would a fruit fly be informative or interesting? Right at that time, in fact, Matt Scott was a co-discoverer of the fact that in these fruit fly genes, there was a little stretch of DNA that turned out to exist in genes in earthworms and butterflies and mice and humans. And it turns out that those same bodybuilding and body patterning genes that sculpt the fruit fly exist and sculpt us. And that was discovered in the 1980s, and it blew everyone's mind. I don't know of anyone who claimed that they saw that coming, but that was the sort of the revolutionary discovery that said, oh my gosh, not only are these fruit flies interesting, I mean, they're a passport to the whole animal kingdom, which meant, yeah, if you wanted to study how to build a mammal, they were a great thing to know about. But if you wanted to understand how did the diversity of the animal kingdom evolve, well, we were staring at the right genes to ask that question. And that was really my driving passion, was the diversification of the animal kingdom. And who knows what lessons there are in there, except for, we often make discoveries that nobody foresees, and they're sort of game changers in terms of people's perspectives. And also individually, if you're really driven to ask a certain question,
Mm-hmm.
I'd be the last person to tell anybody, not to do it, because you got to do it. You got to do what makes sense to you. And, I'm so glad that I went after it.
Yeah, it's interesting. There's almost a parallel I can see between the role that chance and adjacent opportunities play in the natural world, and also in your professional world. So
Yeah.
you were doing immunology, you weren't doing evolutionary biology. You were driven by curiosity, and things just sort of popped up by chance, and you sort of seized on them, just like when you describe the snakes or the other discoveries that I know you've made. There's all of these adjacent possibilities in evolution, and you just sort of tinker what's there, and it opens up this whole new world.
Yeah.
Can you maybe talk about that in the context of your first book, which is really about this whole field of how you build animal bodies?
Well, let me take that, because the question is kind of two-parted, which is there's sort of like the culture of science,
Mm-hmm.
and how does a scientist get lucky? I guess is part of it. And the second thing is, what's the parallel conceptually, sort of, in actually how evolution works. So let me just go to the scientist side of it, the human side of that first. I was going to graduate school in Boston, as you did, and I always looked around at the seminar postings of the other schools, and I would just hop on the T and go hear seminars that interested me. Nobody told me not to do that, but
Mm-hmm.
I did that. And because of that, I only went to things that I thought would be interesting, right? So I used that as kind of a R&D tool, right, of what else was going on in biology at the time. So I was lucky to be in Boston, which was such a fertile place for, almost every flavor of biology was going to come through Boston in a year, right? And anything new and different was going to come through Boston because of the schools that were there. So there's one chalk up, which is, sort of being in the right place matters. And then, as we just described it, I got interested in this stuff and I pursued it. But I can tell you that about 7 or 8 years later, so I knew that I wanted to study evolution. But for a bunch of years, I focused entirely on the fruit fly, because what interested me was the evolution of body form, the number and kind of body parts, their patterning, et cetera. And it was important to get to that. I understood, and others understood, that changes in pattern, changes in form must be due to changes in development, because form is made by development, this
Mm-hmm.
process by which an embryo grows up from a single cell and into a complete creature. So it must be changes in that process that give you creatures that look different. So we have to understand that process in order to understand changes in that process. And I was thinking, okay, I had to go hard on fruit flies to try to find out as many secrets of the fruit, through the fruit fly as I could. But how was I going to study evolution? And one day I was giving a seminar at Duke, and I was scheduled the way academic seminars work. I was scheduled to meet with a variety of people. And one person named Fred Nyhout almost didn't make his appointment with me because he had a pipe break in his house. But then we met, and Fred works on butterflies. And he said, Sean, all this stuff you're discovering in fruit flies, do you think it could sort of explain this? And he shows, sort of a case full of spectacular butterfly wing patterns. And I said, well, I don't know, Fred, but I sure as hell would like to find out. And that's how I got started working on butterflies. That was a case of I meet, one of the few guys, I think in the United States I could have bumped up to, but Fred was a huge expert on butterflies, sweet guy, helped us get started. And that led to asking questions about, okay, where do new things come from? And so we started a program where I was we were comparing different kinds of animals and asking, well, if this animal over here has one set of wings and this animal has two sets of wings, what's different between them? Or if this thing has more walking legs and this has fewer walking legs, what are the differences between them? And then new things. So one of the things that's prominent on some butterflies is they have these eye spots, these beautiful concentric rings of pigmentation that look like eyes, look like false eyes on the wings. And Fred had studied those, and he was studying a species that had them, and we brought those into the lab. And the hunch we played was, well, gee, we were studying the building of wings in a fruit fly, and we thought, okay, same genes are going to build wings in butterflies because wings have been around for 300 million years. They're going to use the same stuff. But what about the patterning of those wings? I wonder if they're doing anything new. So we just cloned out all these genes, and then we said, well, what are they doing in butterflies? And most of the genes were boring. They were doing exactly in butterflies what they were doing in fruit flies, marking out various territories, et cetera. But then one gene absolutely beautifully foreshadowed the formation of the eye spots. And that is that's one of those rare, holy shit, stop the presses moments in the lab where you have no idea. I mean, I had no anticipation of what that result would even look, that we'd get a result like that, or even what it would look like until somebody called me over one morning and said, Sean, this is what I see. And, I mean, it makes your heart race. It makes your mind race because you're like, oh my God, I know this is something. I know this is something. What could it be? And it turns out, we wrote this up in the first half of 1994, but it turned out it was probably the pioneering discovery that really kind of was would teach us essentially the generality of how new physical traits evolve. Mm-hmm. And this was an very old gene, a gene that had been around for more than 500 million years, being used in a new way in those butterfly wings. And that, in a nutshell, was how body patterns were going to evolve. And as we studied more things over the next 15 or 20 years, that theme just kept, and as labs across the world study things, that theme just kept coming back and back. That very old genes sort of learned new tricks in certain species and gave us new patterns. And that was really the root of morphological diversity. And, never knew that we were going to get that first clue in such a visual, surprising, crystalline way, one morning in the lab. But, and when it happens, it's as thrilling as I think, anything that could happen to a race car driver or a musician. I think it's got to, I hope it's on par with that.
Well, the thing that really blew me away when I was a teenager, when I was like 18, and I read your first book, Endless Forms, which is sort of about this whole field, was I said, holy cow, sort of life makes sense now. There's not this inexplicable gap between humans and bugs and other critters. It's all the same genetic ingredients, and you just sort of recombine them and mix and match them in different ways, essentially. And that can generate the actual, the astounding diversity that you see.
Yeah. That's an interesting takeaway you got. So yeah, the book, Endless Forms Most Beautiful, it borrows from Darwin's last line of The Origin of Species. And I was talking about form. I was talking about, well, how does all this incredible diversity of form of the animal kingdom evolve? And by that time, the book was published in 2005. We now knew, we could say that basically there was a common genetic toolkit, these bodybuilding and body patterning genes shared by so many animals. And really, made them different was the way they were used, not whether they existed or not, but the ways that they were used. And you're right, that closes a lot of the gaps between everything in the animal world, to see that we're just a little remodeled, four-legged animal, right? Once you see how to build a mouse, building a human doesn't look that complicated either. Building a fruit fly doesn't look that complicated either. So yeah, those genes gave us really a passport to the making of the entire kingdom, and really a logic, which was, and simultaneous discovery, people were starting to sequence the genomes of all sorts of species. And there was an expectation, for example, that humans would have more genes than anything, right? Because we're so complex. And I remember being at meetings and taking bets, because I was like, no way, man. People are like, it's going to be 100,000 genes in us because there's only 20,000 genes in a mouse, or 15,000 genes in a fruit fly. This would be late 90s.
Late 90s.
Late 90s. Just so human genome comes out.
Yep.
And the number may have been up in the high 20s, but that's come down since then. It's like, yeah. You don't need any more genes to make a human than you do to make a fruit fly or a butterfly, or a snake. You just use them in different ways. And really the double sort of whammy of that, which interested a lot of people, surprised biologists, but then interested non-biologists, because they were like, goodness, we're really, genetically, not only not that different, but not that much of a mystery. Just another four-legged animal in the animal kingdom. But it was those kinds of discoveries that people started asking me, to be in documentary films, or for interviews with the press, or to give public lectures to explain the discoveries and what they meant. And after doing that for a while, that's what led me to write the book. I thought, well, goodness, I could either give this talk, umpteen times, or I could put all these thoughts in some organized way down on paper. And then when the filmmakers came along, I thought, well, film goes even farther, because a lot more people will watch a film, an episode of Nova or something like that, than will read a book. And so, let's do films. And that's what really catalyzed the whole science communication side, was that fortunately I was in a field and directly involved in a lot of cool discoveries, and directly involved in communicating those discoveries. And it just grew from there.
Interesting. So there was never a plan. It wasn't premeditated that, okay, I'm going to become a scientist, run a lab, and now I'm going to start writing books. It just sort of spilled over from your curiosity, and people asked you to talk about it, and then you just sort of formalized what organically happened.
Yeah, and I think it was even somewhat connected to my teaching, because I always enjoyed classroom teaching. I liked the challenge of trying to communicate discoveries and ideas and thoughts to, first timers, right? To students. And that hones a certain, whether it's skills or not, I did my best. That's all I can say. But that at least that challenge is interesting. How do you communicate kind of what's up in your head, or what the scientific community has found out, and how do you communicate that with people in a way that's accessible? I love that challenge, still love it to this day. And that's what all was that was the confluence of things that were sort of going on at that time. But the research was incredibly exciting. New discoveries were happening at a pretty interesting pace in the lab, or in my colleagues' labs around the world. And I was interested in this challenge of how do you communicate this? And no, it was no plan. The original plan. So in 1982, 83, I said, I want to study the making of the animal kingdom. I want to study, how you get snakes and butterflies and giraffes. Okay. That was probably the real root motive. And I followed what I think was a fairly rigorous scientific path of what made sense, find out how some animal does it, and then take that information to studying other animals. But the public side of it, there was no anticipation of that. How could there be? You're lucky in this business to make any discovery, let alone a discovery that somebody finds interesting. And but as I said, I enjoyed the challenge, and I certainly must have enjoyed the feedback that I got. I think a really important audience simultaneous with this, Nick, was 2005, if my memory serves, was the year of the Dover trial on intelligent design. I'm pretty sure.
I forgot about that.
So here, it's to me a golden era in evolutionary science. We've got access to the DNA record. The discoveries are just cascading out of labs. Okay. And we've got this 19th century debate going on of whether or not evolution is real. And so one of the most important audiences I met in this whole time period were biology teachers in large numbers. Like I got invited to conventions. And I tell you, they're the most inspiring group of people to be around. This is essentially this is the frontline army for biologists and for the future of biology and science. These were the people who were taking this to, you know, large numbers of students year in and year out who spent, day after day in classrooms with kids trying to get these ideas across. And I thought, wow, first of all, what have we discovered sort of new that can help them with that? Either make it more engaging or more sticky. And how do we do that? And so that had catalytic effects on some of the other decisions I made after that time. But I saw new ways to teach evolution because we really could go right down to the specific changes in DNA that changed traits. For decades, evolution was sort of described in the abstract sense of, genes must be changing for things to be changing. Now we could show you which genes mattered, which changes and which genes mattered most, and exactly how they added up to making differences. And that was game, set, match in terms of evolutionary process. And, for evolution deniers, it was game, set, match, right? There was no more sort of, gap or uncertainty to try to point to.
Yeah, the subject of not just the denial of evolution by certain people, but the denial of different scientific findings has fascinated me. And it's a really important subject at a societal level, I think. One thing that I found that I still find interesting about it is it's not just one group of people in society that denies science. It's different groups of people denying different forms of science for different
Yeah.
reasons. Can you
Yeah.
speak a little bit about what you think the underlying psychology is around why people sometimes meet with science in a combative way?
Yeah. Well, let me start. So there's a piece, if you don't mind me telling you. So a recent piece I wrote on this, which would be a more organized and thoughtful digest than what I can, verbalize today. It's called the Denialist Playbook, and it's online in Scientific American. And I sort of try to unpack denialism. And let me go right to sort of the bottom line. And I'm not talking about like doubt or so. Look, skepticism plays a big part in science. Let's define denialism. Denialism is really disregarding consensus facts from the scientific community. Okay, so and at the root of that, and denialism and the tactics of denialists are really sort of a rhetorical fog. They're throwing up all sorts of sort of objections and what ifs and yeah buts and all this kind of stuff. This is just a smokescreen for the fact that there is some fundamental philosophical disagreement that they can't surmount. So in the case of a young earth creationist, well, evolution is irreconcilable with their worldview. The world is, the earth is over 4.5 billion years old. All of life evolved naturally with no divine intervention. That's a different worldview than a young earth creationist has. So their only tactics they have left is just to say, what? The scientists are wrong. They've misinterpreted all of their data. Or the scientists are malicious and they are, militant atheists trying to undermine the American way of life. Okay. Or we're dangerous for even saying these sorts of things, or we're taking away their religious freedom. So there's this whole set of rhetorical arguments that are thrown up to try to sort of raise doubt in some or in other, in their community's mind. So that's part of the psychology. Psychology is what happens when you confront facts, consensus facts that are incompatible with a worldview.
Yeah.
Another example, which comes from a totally different world, but it's in the article. And this is where this was for me a formative experience in terms of learning about denialism. It has to do with the story of the polio vaccine in the mid 1950s and chiropractors' response to it. And it turns out that not everyone, but most chiropractors were opposed to polio vaccination. And I thought, oh, wait a second. I'd grown up with this story as like one of the medicine's greatest triumphs,
Hmm.
to thwart this scourge that paralyzed people and killed kids. And in 1955, it was second only to the atomic bomb in terms of what Americans feared most.
Oh, wow.
So polio, as scary as coronavirus, right? At least for sane people. And but chiropractors opposed it. And you're like, well, how do they oppose it? It was a large scale clinical trial. It showed that it prevented the disease.
Yeah, why
People lined up.
chiropractors?
Well, simply put, well, chiropractic was invented in the early 20th century. And the theory of chiropractic was that all disease originated in the spine,
Hmm.
and that manipulation of the spine was necessary to do this stuff. Chiropractic was, generously, you would say at the time, alternative medicine. It didn't use sort of the scientific method. It was never, put to blind clinical trials or anything like this. So, and chiropractors had a pretty good business going of polio patients. So you have people that have,
Ah,
yes, they have spinal problems, they have mobility problems, they have partial paralysis. And so they're going in for spinal manipulations and things like this. So, unfortunately, I think it just sort of boils down to both philosophical and probably a self-interest. The
yeah.
self-interest was it's part of part of chiropractic's practice, but philosophical was they were essentially opposing the germ theory of disease, the idea that microbes like viruses and bacteria were the root of disease, as opposed to misalignments of the
Mhm.
spine. And I thought, oh my God, okay, that's 1955. Okay, let's go 50 years later. A significant portion of the chiropractic community still opposes vaccination, like
Wow.
routine child vaccinations. And when I wrote this article only a month or two ago in Scientific American, I heard from chiropractics on both sides of the aisle. Some who said, "Thank you, you nailed my colleagues to the T." Others who said, you know, what company are you shilling for in writing
Yeah.
such an
Right.
article? And for the record, I was paid zero, which is a typical rate you get in academia. So, but but this is what you hit, and whether it's climate change or it's vaccination or back a long time ago, tobacco smoke and cancer, when you have people who have some kind of interest
Hmm.
in resisting the science or in the science not being true,
Hmm.
you can see these rhetorical, and then it becomes, of course, political, and then it gets, all
Hmm.
sorts of things happen. So um, the really important thing about denialism is to try to figure out how to sort of penetrate that fog.
Hmm.
And fundamentally, you got to ask, why would these people be opposing, polio vaccination? Why
Hmm.
would, well, you know why the tobacco companies would try to
Right.
claim
Right.
that tobacco smoke didn't cause
Well, what
cancer.
about? I
But
think
yeah,
so. The thing about creationism makes sense because there's this deep philosophical, spiritual belief system that runs right against some facts.
yeah,
The chiropractic or the climate change or the tobacco thing makes sense because there's entrenched business interests that run against the science. What
yeah.
about something like what about an upper middle class mother with a child who has autism who becomes a militant anti-vaccination crusader? Where does that come from?
Well, it comes from that experience, because usually the child's autism might be diagnosed right around the time of childhood vaccinations. I've met such parents myself. It's
Hmm.
hard for them to think that it was not caused by vaccination. And of course, it's a very, for it varies, of course, for in terms of parents' experience, but severe autism is a tremendous challenge for parents to deal with. So it's coming from a very understandable experience.
Hmm.
And there are going to be voices and sources out there. They're going to enforce that point. You're going to, support that point of view. This all started in 1998 with Andrew Wakefield's now discredited study in the Lancet that alleged that they saw a high incidence of autism in a small number of British kids um, uh, close to their MMR vaccination, and raised the question, oh my gosh, is autism being triggered by vaccination? But within 3 or 4 years, very large scale studies in the United States, in Scandinavia, and in Europe could not replicate that. Much larger studies than the 12 kids that were in the Wakefield study. And so basically, the incidence of autism is the same in kids who are vaccinated and kids who are not vaccinated.
Hmm.
That's kind of fundamental. But nonetheless, 20 years later, it is very hard to get that thought out of people's minds and to say, there must be something to this. I heard it. There must be something to it. Unfortunately, so I think it's, I have a lot of sympathy for parents who confront this and are confused by all those voices that are out there. So, because a lot of what denialism is sort of just raising doubt,
Mhm.
right? It's, can you really trust the pharma companies? Can you really trust the medical establishment? Did you really feel like you were treated well when you, for example, maybe your kid was first diagnosed? So there's then a lot of momentum to the I don't trust
Mhm.
this group of people, and I have another set of sources that I trust, and those might be vocal, anti-vaccination critics. Take RFK Jr. There's an editorial either in the Washington Post or the New York Times today by his niece, because RFK Jr. is out there against the COVID vaccine again. He's been an anti-vaccination person for 20 years or more, and even though the data is, doesn't support this whatsoever, there are still folks that once they took an entrenched position, it's very hard for people to walk back from those
Mhm.
positions. So a mother that experiences this, or or parents of a newborn for maybe first child, they're like, well, what should we, my gosh, it's a big battery of vaccinations. God, and who likes to see needles go into their baby? So they're hesitant for understandable
Mhm.
reasons. But that's where pediatricians and other kind of public health information comes into play. So I think it's understandable why there's vaccine hesitancy. I think, strident anti-vaccination forces, um, we, it's harder to unpack where they all might be coming from. It might be coming from a personal experience. It might be coming from a wider worldview of sort of distrust of orthodox medicine or pharmaceutical companies. But nonetheless, it was concerning. We made a film about 6 years ago for Nova
Mhm.
called Vaccines Calling the Shots. And we kind of explored vaccine hesitancy for all these conventional vaccines. Okay, so maybe as vaccination rates were dropping, we have more cases of whooping cough, some outbreaks of measles, et cetera. The, the human toll of that vaccine hesitancy seems pretty modest. Now with COVID-19, if a third of the country decides not to get vaccinated, and if that is a point of view that's, replicated around the world, perhaps even worse in some parts of the world, oh my gosh, we got a problem. We're not going to get rid of this thing. We're
Mhm.
not going to get rid of this really nasty virus. And so the stakes now are sky high for
Mhm.
vaccination.
So we're talking about vaccination. We've talked about evolution, and given the very special circumstance the entire world is in right now with COVID, can you talk about viruses? So what are viruses? How are they
Yeah.
different from normal critters? And how can understanding the ways that they evolve and the ways they evolve differently from other creatures, what what can that tell us about how we should be handling the situation today?
That's a great question, Nick. And we're having this conversation right when I think the reports this morning or last night, where's this, new variant in Colorado. What does all this mean? Okay, so viruses are circulating everywhere. You and I've got them in us. That viruses are little bits of genetic material that replicate inside host cells. So these are not things that can exist free living out on their own, like say, for example, bacteria can, or fungi can. So they require a host cell to replicate. And animals harbor a lot of viruses. And if you think of all the diversity of animals on this planet, there are a lot of viruses across the globe. And every now and then, when there's contact between humans and wildlife, is an opportunity for viruses to jump from that wildlife into humans, an event often referred to as spillover. So the
Mhm.
virus is spilling over from a natural reservoir into humans. That's what happened with COVID. That's what happened with HIV. That's what happens essentially every year with flu. It makes a cycle through usually birds.
Oh, really? I didn't know
So
that about the flu.
flu's recombining with viruses in pigs and chickens and other domesticated livestock
Okay.
to get
So
the
every
different variants
year,
of flu every year.
most years it's a spillover event.
Essentially it's a spillover event with flu. Yeah,
Okay.
yeah.
Wow.
Um, so But getting back to something like COVID, HIV is another good example, but it's of animal viruses coming into humans and then being transferred from human to human to human. So viruses, they are small, they are present in large numbers. When a virus replicates in you or I, it makes lots and lots and lots of copies of itself. So with lots and lots of copies of itself, there's lots of chance for evolution, because if you make, a billion copies of a virus, those billion copies are going to carry a mutation here and there. And those mutant variants may have different properties than the parental virus. That's why, for example, this variant is making news, is because it looks like it might be a bit more transmissible than sort of the parent strain of coronavirus. And if it's more transmissible, what's that going to mean in terms of how it moves through the human population? Doesn't look to be more pathogenic, does not look like it makes any more severe or less severe illness. It just may be, for example, binding to our cells a little more readily. And so a little bit of virus gets a foothold more easily than the parental virus. So why is this all relevant to us? We live in a constantly evolving environment, and nearly 8 billion of us are making a lot of contact with wildlife that we didn't necessarily make before, through bushmeat or through encroaching in forests, as we cut down the Amazon, as we invade the Congo, et cetera, et cetera. That's where Ebola is coming from. So Ebola is coming from what we think is a bat host in its repeated cycles of infection, spilling over to humans. So, if you thought that evolution was just some subject that was for, dusty books and fossils and, old natural history museums, no, we live in a world where all sorts of things are evolving. The microbes that we try to treat with antibiotics are evolving. The viruses that are out there in the world are evolving. And as we've now experienced, these evolutionary events can have profound consequences for humanity. I can tell you that 4 years ago, we made a film on spillover and talked about Zika and Ebola and some other things like Nipah and flu. I never imagined something like this. I never imagined that a virus could spread so far, be so dangerous, and essentially paralyze human activity across the globe for a year or more now. I'm also glad that molecular biology has evolved very quickly, and that we could develop countermeasures like these vaccines, faster than any time in history. So, so from a perspective point of view, it's an accident. Random mutations happen in viruses, just as they happen in us. Some of those random mutations change those properties of the viruses, a chance encounter with a human, and we're off to the races.
So you mentioned how fast we've been able to adapt to this virus. We've been able to basically go from 0 to 100 in terms of vaccine development in a very short amount of time compared to historical vaccine development. What is different about how this new generation of vaccines from Moderna and BioNTech are working, and how does that impact our ability to try and keep pace with viral evolution?
Yeah. Great question. The way vaccines usually were made, let's say in the lowest tech era, were often just a killed form of the virus. So you'd grow the virus in vats of tissue culture in a lab. You'd inactivate it chemically or with ultraviolet or whatever it might be. And you inject that into people. So it's not able to infect them, but they're basically seeing the killed virus stimulates their immune system enough that they have protection in case they see the actual live virus. Those vaccines, which we use for lots of things, so flu is an inactivated vaccine, have generally worked okay. But for some viruses, they're very, very difficult. AIDS, we still don't have a vaccine because that virus mutates so quickly that basically there's no virus you could immunize people with that would protect everybody against AIDS. So usually because the way we conduct the clinical trials of these vaccines, there's a fair amount of sort of patient bureaucracy with this, in that, you take one step and then it gets sent off to the FDA and it takes months to review, and then you start the next step. What happened was all those things were compressed. So basically the federal government stepped in and started funding development of the vaccine or the advanced stages of the vaccine before the other stages were sort of closed, so that these manufacturers could be scaling up the production of the vaccine before they even knew whether it worked. Otherwise, it'd be huge financial risk. If you start scaling up a vaccine before you know it works and you find out it doesn't work well, you may
Mhm.
have lost $1 billion and your shareholders are very unhappy. So basically, the federal government backstopped a lot of these vaccine developers so that they could be scaling faster, so that they could jump into clinical trials faster. But there's also a technology shift. So
Mhm.
instead of killed viruses, Pfizer and Moderna, or BioNTech and Moderna, use a technology that's never been used for large scale vaccination, in that they are actually putting messenger RNA, which is a form of RNA that carries the genetic information for making one piece of the virus. They're putting that in little lipid droplets, injecting that into your arm, and your cells make a little bit of the virus that then primes your immune system. So these are mRNA vaccines, again, never been used at scale. And goodness, nobody could have predicted they would be as effective as the data turned out to be. Another thing about science, everybody's got to keep in mind, you don't know the results until you actually do the experiment.
Mhm.
And for Moderna and BioNTech, Pfizer, it just turned out really well. So there was a technological change. And the thing about mRNA vaccine was that as soon as those viruses were sequenced in China from patients,
Mhm.
we could in the lab here make those molecules very quickly and get them tested in animals and start right on the course of vaccination.
Mhm.
So everything was sort of done in hyperdrive. Now, just today in the UK, a different vaccine made by Oxford AstraZeneca was approved. And this is, again, technologically something different, which is a little piece of the coronavirus, the same part that's being used in the mRNA vaccine, is in a vector, another virus, a harmless virus called adenovirus 2. And this is a virus that can't replicate in our cells, but acts as sort of a shuttle to get that genetic material into our cells, making a little bit of the virus so that our immune system gets primed to it. So it's ready to go in case we encounter the real virus. So both of those technologies are ways of delivering the sort of important part of the virus that is involved in infecting our cells. So this spike protein that's on the
Mhm.
outside of the coronavirus, that's the sort of the key that unlocks the portal on our cells that gets the virus into our cells. And if you block that key, it can't get in. So that's the rationale for vaccine design. But researchers were working on coronavirus vaccines for things like SARS and MERS. They had some years of experience with these technologies, so that when this coronavirus came along, they said, hey, you know, let's see if we can make a vaccine this way. So it wasn't like the research did not exist, but really it was benchtop research for other coronaviruses that they then said, well, let's go for it. Let's see if we can make an mRNA vaccine. Let's see if we can make an adenovirus-based vaccine. Another technology, which is just to make the spike protein as it is, that's the strategy being used by a company called Novavax. They're
Mhm.
just entering phase 3 trials now. That's very similar in sort of design to something like the hepatitis B vaccine, which is pure viral protein. So that's a little later than these other vaccines. But this reflects, you know, the biologist tools for doing these things have changed a lot over the decades. And so we had more tools that worked a lot faster, so that lots of folks jumped in. I mean, this was a worldwide effort, and there's, lots of vaccines under development. Um, but because of the societal imperative, the financial backstopping of these companies so that they could start making tens and tens of millions of doses before they even knew the vaccine worked. That was really important.
Mhm.
Yeah.
So I've heard the new mRNA vaccines described as programmable. What does that mean, and what would it allow us to do if, let's just say tomorrow, there's a brand new coronavirus strain, brand new
Yeah.
mutation, the spike protein's now completely different, and the vaccines we have right now aren't as effective? How quickly could a new vaccine be developed?
Well, I mean, it's just going to be a speculation. I think it was 60 days for the first one
Mhm.
that essentially the vaccine was formulated. So I think it's probably 60 days or less that we'd be able to spike. I shouldn't use the word spike. So right now, the mRNA that's in there is for essentially the prototypical strain of coronavirus. If we thought now that coronavirus had evolved enough that we wanted to have some variant or combination of variants represented in that vaccine, we could add the mRNA for those variant proteins to that mixture. Same technology.
Mm.
So I think that adding a variant to the Pfizer vaccine or the Moderna vaccine would be pretty darn straightforward. Now, I don't know, the regulatory authorities might want a new clinical trial to make sure that that was
Mhm.
protective. Or it may be, again, just because of the imperative, because we're in the middle of a pandemic. Realize most studies are done at a slower pace because the urgency and the societal impact is not that great. That maybe the regulatory authorities would allow use of that vaccine, essentially piggybacking on everything that was established about the first vaccine. They'd still want to probably gather data in the field, but they might allow the vaccine to go forward without all the steps to the trial. Now that essentially the principle's been proven, for the mRNA-based vaccines. So we would have a response, and the adenovirus is also programmable so that we could quickly be expressing that variant. So I think it's 60 days that we'd probably be working on it. Whether or not the regulatory authorities would allow that to go into people in large numbers without some formal testing, I can't speculate. But if this was something, imagine this: if 2 or 3 months from now we found out there was a vaccine-resistant strain, a strain that we didn't think was starting to circulate, that was not protected against by the vaccine. Man, I don't know. That's a really tough ethics call for regulators, which is, the world is really suffering. And would you take that chance? Well, I hope we don't have to find out, man. I hope we don't have to find out. But, we feel good that we can respond on this time scale. At the same time, every month going forward, you can see what the death toll and what the illness and the economic toll is going to be. So, every month really matters right now.
Yeah, it's amazing that the science is at a state where we can actually move this fast. It's absolutely amazing.
Yeah, it is amazing. And there's one other part which is probably unseen by the public. So they're getting the news reports that say, the phase 1 data that came out, I almost can't remember, probably midsummer, that said, look, when you put these vaccines into people, their immune responses are similar to people who got the real virus, got infected and went to the hospital. Then the efficacy data surprised everybody. I mean, no scientist is going to say, yeah, I'm going to make a 94, 95% effective vaccine out of the shit. A lot of the vaccines we have aren't 94, 95% effective at preventing infection. That's just the way the biology works. So, they hit a grand slam first time at bat. That's pretty remarkable. So I think, the scientific community has a lot to be proud of, but the invisible part is that, the sharing of data and the sharing of studies, because a lot of stuff is going to, a lot of scientific studies of the coronavirus are going to these preprint servers before they go to journals. This is an innovation that happened in science publishing over the last few years to sort of speed up the otherwise somewhat slow pace of formal scientific publishing. Now coronavirus hits and people are just sharing their findings, before peer review. There's some risk to that, but the upside of that is that the information is getting into each other's hands much, much faster. And man, I've tried to, penetrate the mass of that literature when I go ask a question and say, well, what do we know about patients' response, 3 weeks in? And I find all these papers from all over the world, and it's just happening at lightning speed. So the ability for scientists to share information is unprecedented. And the amount of sharing that's going on globally is unprecedented. Everyone's trying to learn as much about this virus and our responses to it and what works and what doesn't work as fast as possible, and sharing that openly. That's a pretty remarkable thing. Sure, we'd love to know more. We all, we wish we knew in March what we know now, and I'm sure we're going to know things this coming March that we wish we knew today. Look, but nonetheless, the sort of everything about the scientific enterprise is moving at a speed I've never seen in my lifetime. on the coronavirus in particular.
Yeah, it's amazing. So we've talked a lot about the role that chance plays in everything. So why don't you walk us through the new book and what it's about and what inspired you to write this book now?
Yeah, well, I wrote it pretty much pre-pandemic, but it was in editing when the pandemic came out, so I got to update that a little bit. So it's called A Series of Fortunate Events. It's about chance in the making of the planet, life, and you. Why would I take up a book whose theme is about chance? And it's because I think it's an underappreciated force in the world. And the book in particular looks at the sort of geological, cosmological, and biological accidents that make us us, and that sort of the astonishing series of things that had to happen for you and I to have this conversation, for you and I to exist, for us to collectively to exist, for us individually to exist. And a bunch of stuff that's been only discovered in the last 50 years, that scientists go, wow. And it's sort of some of the fun of the book, and I try to make it fun. I guess that's almost a pledge to anyone who decides to read it. Shortest book I've ever written. Trying to make it as digestible and as accessible. But you can almost think of it as a bunch of what ifs. And the first what if I start with is the asteroid impact 66 million years ago. A lot of people have heard about this. Asteroid hit the Earth 66 million years ago, and you probably know, wiped out the dinosaurs. Also triggered a mass extinction and wiped out 3/4 of all plant and animal life. Well, why is that a big what if in terms of our point of view? Well, those dinosaurs and those large reptiles had dominated the Earth for 150 million years. Mammals were a real sort of minor player on the stage at the time. They'd been around a long time, but they were generally small, not too significant part of the ecology of the planet. But after you cleared the world of the dinosaurs, after that asteroid, mammals took off, and they got, in a very short period of time, much larger than it had ever been, and they became the largest animals in the oceans and on land. And some of those mammals evolved into primates, and those primates eventually gave rise to us. So had that asteroid, now let's add a little color to the asteroid. It's a 6-mile wide rock that hits the Yucatan Peninsula 66 million years ago. Well, that rock may have been circulating the solar system, I don't know, maybe 4 billion years. And at one particular day, 66 million years ago, it enters the atmosphere at 50,000 miles per hour and slams the Yucatan. If it enters, well, let me tell you a little more about the Yucatan. We now appreciate that the Yucatan had the kind of mixture of rocks, sulfur and carbonate containing rocks, that when you vaporized all that stuff and shot up in the atmosphere, it made a really toxic stew, as well as deflecting sunlight. So basically the world was in a blackout for probably 10 to 30 years, no sunlight or very little sunlight. So plants collapsed, everything that ate plants collapsed, everything that depended on things that ate those things that ate the plants collapsed. That's why you had a mass extinction. But only about 1 to 13% of the Earth's surface has the right combination of rocks that when vaporized could cause that mass extinction. So had that asteroid entered maybe a half hour sooner, it lands in the Atlantic, probably no mass extinction. Half hour later, it lands in the Pacific, probably no mass extinction. So that's a pretty big what if. If that rock does not hit the Earth, or if that rock hits somewhere else on the Earth might still be a reptile-dominated world. Mammals are a minor player. Humans never come along. And I hope that's a little mind-blowing for people to contemplate. That had we not been thrown this fastball from the cosmos, we don't exist. Marvel Comics don't exist. Everything else that everybody depends upon does not exist. And so the book has some of those stories, both sort of our collective existence. There's another big collision that involves the Indian subcontinent. Without it, we don't get the ice ages. Without that, we probably don't get large-brained humans. But even right down to you and I. So let's play a little pop quiz. You're good at math, so you'll get this. But anyway, how many genetically unique children could any human couple have?
Well, I cheated because I read the book. But if I try to go on my raw intuition, what do I think I would think the answer was? What do I think the average person might think? I don't know. You might guess dozens, maybe.
Yeah. 23 chromosomes was coming from dad, 23 chromosomes come from mom. So maybe it's 23 or 46 or 92, but it's actually more than 70 trillion. And so that means that no two of us will ever, unless there's an identical twin, no two fertilized human eggs will ever be identical, ever. We are each unique. So we can, cherish that, right? It says, but that's because of just the random assortment of chromosomes in fertilization, and the sort of the sperm race. We're just, 1 out of 100 million sperm that fertilizes the egg. And so each of us is essentially a biological accident, a happy accident, hopefully, but a biological accident. So the book is all about sort of the role of chance in our existence and in, as I said, in the way that the planet is. And I hope it sort of builds in people's minds this understanding that we live in a chance-driven world. If everything you look around, and chance deep within every organism, the process of mutation, which gives us all biological diversity on the planet, is a chance process. That's how fundamental chance is to the biosphere.
One of my favorite stories in the book is in the beginning, in the introduction, I think. And you describe a scientist that most people probably have not heard of, who's very famous among biologists. And this person said that the purpose of science is not to make new technology. So we just had a whole discussion about how amazing it is that science creates so much amazing technology. But this guy said that's actually not the primary, not the essence of science. So who was that person, and what was his answer to what science is really for?
Yeah, so this is French biologist Jacques Monod, Nobel laureate in 1965. He was a pioneer of molecular biology. But as a Frenchman steeped in his country's philosophical traditions, he was interested in what discoveries of biology had to offer sort of to the philosophical discussion of life, and felt it was really underappreciated, particularly at the time he was living, in the late 60s and early 70s. He felt that, and felt strongly enough, that he wrote a book called Chance and Necessity to try to bring these discoveries of biology to a wider audience. Now, it's pertinent, at least in my telling of the story, that he had been friends with Albert Camus, writer, philosopher Albert Camus, who was also interested in sort of, is there a purpose in life? Why are we here? And when they met and became friends, he now had, one of the great living biologists of his time as kind of a personal consultant on all matters biological. But what Monod came to the conclusion of, and this came directly from understanding molecular biology, that this process of mutation is random, and therefore the source of all diversity in the biosphere, all variation, all complexity, all beauty is chance. And he saw the philosophical power of that, which basically it vaporizes all notions of design and intent in the world, which is really, 2,000 plus years of Judeo-Christian thinking. So, but remember, these are facts from biology. So the philosophers were riled up about Monod because he was sort of infringing on their territory, but he was bringing new information and saying, hey, we biologists say, look, biology is driven by a chance-based process. What does that mean? What should that mean to the world? So it prompted him to write this book, Chance and Necessity, and it was a book I read as a teenager, and but I felt that still chance was not as widely appreciated or as deeply appreciated as it should be. And it's Monod's book was 50 years ago, and he passed away only a few years after he wrote it. And I thought, okay, well, maybe it's time to revisit that with new discoveries. Monod didn't know about the asteroid impact. He didn't know about the plate tectonics that had reshaped the world. He didn't know necessarily the connection between mutation and traits like we know today. He didn't know about, for example, the origin of cancer. So I thought, let's update our thinking about chance with discoveries from the last 50 years. And I have spent a lot of time in sort of Monod's trail. So I do this humbly, offering this little update on our view of chance.
So I love the book, by the way. So it's highly recommended by me.
Thanks,
I
Nick.
think you've written a number of books now. So you're a best-selling author. You're obviously a successful scientist. I want to get into the filmmaking stuff in a little bit, but I want to talk about actually maybe the very beginning for you. So even before you got into science and before you were letting your curiosity drive where you were going once you were in the formal scientific world, what were you interested in as a kid? What were you studying in college? Was it always science oriented? And what was that like early on?
Okay, well, I think I know my origin story. You never know, is it if it's accurate, but I'm pretty sure this is accurate, what I can start with. And it's important. I gave you a place, right? I grew up in Toledo, Ohio, on a city perched on the edge of Lake Erie, a lake that I never swam in or ate anything out of my whole childhood because it was notoriously polluted. It is not a hotspot of biodiversity. So as a little kid, and I grew up on a city block which had 9 houses on each side. My whole neighborhood, I went on that block after block after block. So squirrels and pigeons were, and an occasional robin was the only wildlife in my backyard. But on the edges of the city, if I got out into the woods, that's where I found frogs and salamanders and snakes. And that is what got my juices flowing. You'd flip over a log, you didn't know what was there. Oh my gosh, look at that. Look at that. That's so cool. Look at that creature. Oh my gosh. And they were just, to me, fascinating. And coupled with, at the time, there was a show on TV called Mutual of Omaha's Wild Kingdom, hosted by Marlin Perkins, who was the curator of the St. Louis Zoo. And they went around the world to, fascinating places and looked at wildlife. And I just thought that was captivating. I know how much, Africa, the Amazon, the whole world was an amazing place. But I was in this rather domesticated place of Northwest Ohio. And so it was log flipping. And I know this experiences of a lot of other biologists that we discover in each other that, if you liked butterflies as a kid, or you liked fossil hunting as a kid, or maybe you've lived on a farm and you liked livestock and animals, that's roots of a lot of biologists that I know. And for me, it was reptiles. And I just thought they were fascinating creatures. And I thought, okay, I'll study biology in college. Now, did I have any idea where that was going to go?
Mm-hmm.
Oh, hell no. What do you know? I mean, you've got to think back to yourself. What did you know at the beginning of where you might go? But what happened was I got taken into a lab early. Sound familiar? And I got to see up close how science was done, and then got invited to do some of that science. And I loved the lab work. I loved the ability to design an experiment and then see what would happen. And so I became then an indoor biologist. So even though I had this great love of wildlife in particular, I became an indoor experimental, kind of molecular biologist type, because I loved answers at that level. I still do. I love getting down to the absolute, fundamental mechanism of something if you can. But as a kid, it was the thrill. And I think about this a lot, Nick. It was the thrill when you flip over a log. Most of the time, there's nothing under there, or it's just worms and millipedes, right? Mm-hmm. But when you find a Jefferson salamander with this shiny black body with little blue dots on it, I mean, that's like finding the Hope Diamond, right? It's a huge visceral thrill. When you're just out, driving along a road, oh my God, there's a snake in the road, and you hop out and check out this, beautifully colored milk snake or something like that. That's what made my heart race. And I didn't have a clear idea what I could do with it, but I decided I wanted to study life and biology as an undergrad. And then I had enough people take me under their wing to sort of say, okay, this is how you become a biology student. This is how you then become sort of an apprentice biologist. And, this is what we do. And I thought, okay, I'm going to roll with this.
Yeah. Well, I mean, a common thread here is that you've always seemed to be curiosity driven first. And we've talked a lot about the role of chance versus planning in your trajectory. So what advice would you give to someone generally about how they should look at seizing chance opportunities that arise throughout life?
Heavy. I tell you, I've been in this position a few times, Nick, where I give advice and then I get an email later that says, yeah, I'm giving up engineering for art. And I'm like, oh, uh-oh. Just don't tell your parents where you heard that. Look, full disclosure, my wife, Jamie, and I were parents of 4 kids. That's probably the closest, you know, those are people closest to me in life. And what advice do you give a child? And it's actually the advice I got from my parents, and so it's advice I gave my kids, which is my parents just said, find something you love. I was the weirdo in my family. No scientists in my family, a lot of musicians, but I went out in the backyard and looked at stuff, and they thought, well, he's happy, let him do it. And my mom let me keep snakes in my room, and let me keep night crawlers in the family refrigerator. And even though she was grossed out by them, she let me do that. So we said the same thing to our kids, and it turns out they all went into the arts. So I have to believe that, and I truly believe for happiness in life, you have to pursue those things that arouse your passions. You just have to. It doesn't mean you don't get to do it all the time, all day long. Most all the artists I know, including my kids, there's some balance where there might be a day job that helps pay the bills while they work on a new song. But nonetheless, they have to be doing these creative endeavors, I think, to be happy and to be fulfilled. And so you're asking, what advice do I want to throw out there to people? I think if you can find something that you find fulfilling and can figure out some way to make a living at it, you win. I think if you can, I remember asking my youngest this. I said, how much of your day, this is post-college, I said, how much, he's a musician, how much of your day do you get to spend immersed in music one way or another? And he said, well, more than half. And I said, man, I think you're beating the averages. Most jobs, most endeavors, no matter what you find yourself doing, there's a certain amount of, administrative work or, time spent that you don't really relish. It just comes with the territory. But if you can spend a fair amount of your time doing things that keeps your juices flowing, keeps your passion up, keeps you, motivated, whether that's working for a cause or pursuing the arts or pursuing science, whatever it might be. I think that's what we're trying to find for ourselves, hopefully in relatively the first half of life,
Mhm.
first 25 or 30 years. If you can find it, then I think you're fortunate. And if you can make a living at it, then you're doubly fortunate. They don't, as you know, they don't go hand in hand.
Well, you've had a really interesting way of figuring out how to pursue your interests as a scientist, but then also tell those stories in book form, but also in the form of films. So talk a little bit about what you're doing for HHMI. What is HHMI and what is your role there, and how does it tie to filmmaking, and how did that happen?
Yeah, everybody kind of hold on for this talk. All right, so I'm head of science education at HHMI. HHMI is the largest life science philanthropy in the United States, supports both basic research and science education. I run their science education programs. I have for the last 10 years. I was motivated to do that a lot because I think the teaching of science is really important, and that I thought there was some way I could contribute to both the content that's available to teachers, and to even the training that's available to teachers. And we're involved in both. But I'm also interested in the public communication of science. And so one of the things I did when I got here, with my trustees' blessing, was to start a documentary film unit. That came from experience of being in a fair amount of films before that, and meeting a fair number of filmmakers, and feeling that a model that had not really been explored is to have scientists sort of right up there in the cockpit with filmmakers telling these stories. Generally, we were subjects that were visited by filmmakers. They did their shooting, they went back and, edited these films, and then, we'd see them later when they were broadcast. And it seemed like a missed opportunity. In fact, myself and a lot of my colleagues were not necessarily very happy with the product, nor were the filmmakers. And I thought, I just kept asking my, you know, these filmmakers I came to know, you know, what was stopping you from doing your best work? And, you know, was there a different model that we could pursue? And so as a science organization, we started this documentary film studio and brought in people from the industry. So my team is very seasoned filmmakers, seasoned industry people, some journalist folks. And what we're basically do is tell stories about science and scientists on film. Now, some of those things we do for the classroom, we don't need to necessarily talk about that today, but the things that your audience can see are, you know, IMAX films for, you know, science museums, broadcast films that you'll see on, you know, PBS or Netflix or Smithsonian Channel, things like that, and different styles of films, more theatrical films that are a little more emotional and inspirational, as well as sort of more informational films about things like vaccination or spillover diseases and things like that. But the thing about film is it's the most immersive medium we have. It travels the globe very well. And a film can be seen by very, very large audiences. Also, film can allow you to do things that's very hard to do in other media. It can help you imagine the invisible or things that are sort of beyond the human scale of experience, whether it's the cosmos or, you know, life inside a cell. It can let you go back in time, you know, recreate things with actors and all that. So, you know, and it's a popular form of entertainment. So science has a lot of stories to tell, and we just have to be good enough at telling them that people want to hear these stories as much as they want to, you know, hear stories from other fields of human endeavor. So I don't know if that gives you that in a nutshell, because anything longer than that would be a long time, because that's a lot of evolutionary steps to go from, you know, starting to write books to working with actually some of the best filmmakers, documentary filmmakers in the world, to tell some of these science stories. So, and look, the other thing I just want to underscore is the craft side of that. These storytellers, their ability to tell a story visually, I'm astounded at times, moved. I remember there's one of my books, Serengeti Rules, was adapted to film, and filmmakers decided that they wanted to sort of tell the origin stories of a few of the ecologists that are in the film. And I was visiting in London. The producer was in London, and we went to a screening room around the corner in a London theater just to see some early work, some early cuts. And I watched it and I said, oh my God, every one of these scientists is going to have a tear going down their eye, because they captured what you and I have been talking a lot in this past hour. They captured that passion. They captured that curiosity. They captured, why would somebody go out in the world for 50 years into a remote part of the planet and just devote their lives to asking a question and understanding how nature worked? And they caught it, and they conveyed it. And that's, you know, that's a talent that is scarce in general, but when paired up with, I think, with some scientific aptitude, it hopefully it's a potent mix. Film won an Emmy, by the way, for best nature documentary. So I'll just, you know, give credit where credit's due.
And what was the name of that one again?
The Serengeti Rules.
Serengeti Rules.
Screening on PBS right now, on PBS's Nature. It's screening for free, so if you want to check it out, go ahead. But it's that's, to me, is a good example of combining the art, the visual art and storytelling with a substantive and important scientific story. Another one I'd throw out there, which is also streaming for free, is called The Farthest. It's the story of the Voyager missions to the outer solar system, launched in 1977. Also won an Emmy for best doc, and it's a hell of a story. And again, great filmmaking with a great story. It's you know, these are they're kind of the equivalent of the eureka moments in the lab. Sometimes things come together and what happens is better than you could have imagined.
One of the last things I want to ask you about is, you know, we've talked a lot about chance and opportunity and creativity and spontaneity in many ways, but at this point you've written several books. Let's just talk about the book writing process.
Yeah.
You must have some kind of creative process. So your last book, you know, you're going to write it you're in the middle of it, you know you've got to get X chapters out. Paint us a picture of what does it look like when you've got your sleeves rolled up and you're doing the work.
Yeah. So when I know I'm gone for it, so that was a book that probably incubated in some embryonic form for 5 or 6 years before I
Mhm.
tackled it. I thought there was something there that I wanted to do, but I couldn't quite see enough of it to do it. But once I saw enough of it and kind of knew stylistically some things I wanted to do, there's a little lightheartedness in the book. You may have noticed. You know me well enough to know that I would be throwing off one-liners from time to time, but I haven't necessarily done that a lot in books. Um, so once I got in sort of, I think, the right headspace for telling the story the way I wanted to, I'm on my back patio on a summer morning at 6:30 in the morning. I wake up while everything's fresh,
Mhm.
um, get to a quiet place, and if I can write for 2 or 3 hours with no clock, with no timer ticking. So like the worst thing for me is like I got to be somewhere at 8:30. Oh, I
Mhm.
have 2 hours.
Mhm.
Because what if it's not flowing? Then you're like, oh, now I got 1 hour. Oh, now the morning shot. So you need enough buffered time there that if it's flowing, you can just
Stick
go
with it.
all the way. You can stick with it and get it all out until you're just like, okay, I'm out. Now I can go off and go to the day job. Um, but it's that kind of process day after day, and I need to keep my momentum up. In other words, I can't take many days off from writing. It feels like the rock slides back downhill on me.
Mhm.
That I'm right there, and then you hit like a knot or something. You're like, God, where am I going to go with this? This way? Am I going to go that way? And you also have to be patient enough that you'll go somewhere, you know, with the story, and it's not working, and you have to tear it up and back up and go another way, and not say, oh, that's 2 days lost. That's just part of the game. But I think that momentum's really important. That headspace that you're in for me is really important, and maintaining that. Um, it's not a trance. It's like a very soft trance. You start to become unaware of the passage of time. You're deep in the story. You're kind of in your storytelling voice even,
Mhm.
and you can feel that you're not getting interrupted. You're not answering the phone. You're not looking at email or anything else like that. You're just in your storytelling voice, and you're just trying to tell the story as engagingly and as clearly as you can. And and to do a book length project, boy, you got to keep that up, for months and months and months.
Hmm.
I don't know if
You
that gives you a little glimpse.
Yeah, no, that's great.
For another book that was 3 years, and
Yeah,
that's
yeah.
that couldn't be. I couldn't do it day after day for 3 years. This book was manageable in probably about a year's time.
Okay.
Yeah.
You mentioned your lightheartedness, and that's something I've always appreciated about you. I There was something I read in the last book that I didn't expect, except once I read it, it made perfect sense. You mentioned stand-up comedians, and you talked about this weird, or maybe not weird, similarity that stand-up comedians and scientists have. So what is that?
Okay, so I was realizing, so I love stand-up comedy for whatever reason. I love a good laugh, and uh, especially I have two boys living in LA, and you go to LA, and all the comedy clubs have just top line acts. And I live in DC now, and I can, in pre-COVID, could go to lots of comedy. So I go to a lot of stand-up comedy and developed, as fans of all sorts of people. And I realized Lewis Black, Sarah Silverman, Bill Maher, Ricky Gervais, Bill Burr, on you go. They deal with, for example, religion or sort of the accidental nature of life in their acts. And I thought, first of all, maybe it's why I like them. But second of all, it's kind of brave because it's not an area that people are comfortable with. They're pretty explicit about saying, hey, this is all there is. There's nobody in charge. And I thought, well, why is this? Why did Eric Idle's another? And so I reached out to some folks, including Eric Idle of Monty Python, who answered. I had a bunch of questions, and I get to communicate with one of my all-time comedy idols. And I just wanted to get at this of why is it in their act, and why, if I think of all people in all sorts of walks of life, it seemed like comedians, there were a disproportionate number of comedians that saw the world as chance driven, as accident predominating. Seth MacFarlane's another. And there's a story in the book about Seth, who was by a hair's breadth missed a plane on 9/11. And Eric came back to me and said, it's about truth telling. Eric Idle. And I thought, holy smoke, how can I get to the age I am and not understand sort of? And then I'm reading these interviews with Gervais and Bill Maher and truth telling just keeps coming up left, right, and center. Now that I'm alert to it, I'm like, yes, they see themselves as truth tellers. And of course, well, that's what we scientists see ourselves as. So we're trying to find out the world as it is, not some construct of a way we wish it was, but how does it work? What is it? What does that mean? Okay, we're here by accident. Okay, there's nobody looking out for us. There's no preset, purpose or whatever it might be. And I'm finding all these comedians who think that way and who reflect that in their work. And so that discovery of comedians and scientists having truth telling as their common modus, I just thought it was fun to explore that. First, it was fun to me to sort of quote discover it for myself anyway, and then to explore that. So the last chapter in the book is this conversation about chance, which is it's a synthesis of thoughts from dead people like Kurt Vonnegut and Albert Camus and Jacques Monod, comedians who had time to answer me like Eric Idle, other interviews from folks like Ricky Gervais. And because when you get into this area talking about chance, inevitably people ask, well, if it's all chance, what purpose is in their life? What meaning is there in life? And I thought that readers would want me to go down that road. So I did in the last chapter, but I did it with these assembled guests that I wanted to have a conversation with, which was Camus, who takes the deep philosophical approach, Vonnegut, who takes kind of the satirical literary approach, Gervais and Idle, who take other approaches, and hopefully that conversation was interesting. I had fun stitching it together.
Yeah, no, it was a lot of fun to read for sure. So last few minutes here, why don't we just close out one more time? What's the latest book? What's the latest film that's out there? And where can people go to learn more about you and what you're doing?
Oh, goodness. Okay. So the latest book is A Series of Fortunate Events, Chance in the Making of the Planet, Life, and You. And I've given you that information, including there's a 2-minute animated trailer for the book, which I think is a good way for people to sample it and see if it tickles their sensibilities, then hopefully the book is worth it. Films, we mentioned, I think the Serengeti Rules and The Farthest would be two sort of exemplary films that have come from our studio. Right now, there's also a film on demand. If you look it up, it's called Oliver Sacks, His Own Life. It's a documentary about the life of Oliver Sacks, very powerful, directed by Rick Burns. So that's one of our current releases that's out there. The theatrical release was affected by COVID, but it's being released in a mechanism that helps support theaters at the same time, making it available on demand. And more stuff, gosh, if you really want to find anything more about me, my website is seanbcarroll.com, and there'll be little bits there about past books and ongoing film activities and things like that.
Well, Sean, thank you for taking the time. I really appreciate it. You've been, for like 15 years now, you've been my scientific role model. You've also been my facial hair role model. And I think your books are a lot of fun to read. They're super interesting. And I think people are going to really enjoy looking you up.
Well, thanks, Nick. I'm glad I at least had the judgment when this kid came into my office and stunned me by telling me he had read Endless Forms Most Beautiful and wanted a slot in the lab. That 4 years later, you proved, an immensely talented, hardworking, incredibly thoughtful scientist, did a great piece of work as an undergrad, which is very rare as it is, great and influential piece of work as an undergrad. So, let's keep having these conversations.